Okay, so that's the second. We've talked about sexual recombination and we've talked about exotic incursion. That's 2 of the 5 sources of variation. Number 3, migration within an epidemiological zone, right? So if you look at Australia, we know that as an epidemiological zone, we know that we have 2 cereal growing regions, an eastern one and a western one, and they're separated by about 1,500 kilometres of desert. There's no wheat grown in here, okay? What we find is that it's not unusual for us to get exchange of rust inoculum from Western Australia into Eastern Australia. So since 1988, we've seen 7 examples where we've found a new pathotype of rust here, and soon afterwards it's ended up in Eastern Australia. So you can see that that's what all these arrows represent, those 7 examples of movement of rust isolates from Western Australia into Eastern Australia. We've only found 1 example during that time where we've had the reverse case, where it's gone from east to west. And we think the reason for that is that the prevailing wind patterns are from west to east. So these rusts are being blown on the prevailing winds several thousand kilometres from west to east. Rarely do we ever see them go back the other way. When they do, it's probably again this human-mediated transport of rust. Okay? What this means in terms of controlling rust for growers is that we have differences in the virulences, differences in the pathotypes between Eastern and Western Australia. So we have many virulences, for example the ones I've shown here, that occur, virulence for these resistance genes are present in Eastern Australia but not in Western Australia. All right, so, and I can give you a very good example of what that means with respect to this gene here, YR17. Okay, so there's one variety in Australia at the moment called mace, a wheat variety. Very popular, it became very popular in Eastern Australia, but we had a mutation to virulence for YR17. Mace became highly susceptible to stripe rust and farmers stopped growing it. In Western Australia, last year, mace was about 48% of the acreage. And this year they say it's going to go to 55% of the acreage in Western Australia. It's still resistant in WA, in Western Australia, because they don't have that virulence there. But it's not a good situation to be in. We shouldn't have that much of the acreage occupied by a variety protected by a single gene. But anyway, knowing the distribution of the pathotypes between those 2 regions and knowing a bit about the migration of the rust between them helps us with telling farmers what the risk is or the threat is to different varieties if we know what resistance genes they carry. Okay, so that's number 3, that's migration within an epidemiological zone. One other example I can give you. This is an example with leaf rust, and it's actually not just Australia but also New Zealand. All right? So you have New Zealand down here, and you're looking at about 6,500 kilometres from this point all the way across to here. In 1984, not long before I started doing the race analysis for leaf rust, we found one of these exotic incursions of leaf rust down here in Victoria. Within 10 years, that pathotype or a derivative pathotype had spread to all wheat growing areas in Australia and to New Zealand. So it only took it 10 years to spread throughout this entire region, 6,500 kilometres. And if you put that in the context of this part of the world, that's the distance you're looking at. All right, 6,500 kilometres. Now I should qualify that by saying that the wind patterns here are very different to the wind patterns we have in Australia. So you're not just talking about distance, we're also talking about wind patterns. But just to put it in the context of the distance you're talking about, took 10 years for an isolate to cover that area just through wind and possibly through human-mediated transport across here. That's the distance you're talking about. Okay, number 4, mutation. And this is the one that I said is probably the most common mechanism generating variation in rust pathogens around the world. Certainly for us in Australia, where we don't have any sexual recombination at all in any of our wheat attacking rusts, we know mutation is the #1 generator of variation. Estimated rate of mutation, about 1 spore in 10 billion has been calculated. You'll get a mutant in. Mutation is a random process, all right? These are just random mistakes that are made in DNA when the DNA is being replicated. That's what mutation is. So you think, well, it's only 1 in 10 billion. I mean, that's not much. But it's also been estimated that 10 billion spores are produced by an acre of wheat with only 10% infection. So if you've got a 1 acre plot of wheat, it's only got 10% infection of rust in it, you've already got 1 mutant. So then extend that out over a huge area and you can see you are going to get quite a few of these random mutants being generated. To show you, and so the message for that, and I'll come back to this a bit later on, is that the more rust you have, the more mutants you have, and the more mutants you have, potentially the more pathotypes you have. So one of the really strong extension messages we give to growers is get those rust levels down. It's good for reducing yield loss in your crop, but it's also good to reduce the opportunity for the pathogen to generate variation. Now, to show you what I mean by mutation, okay, I'm going to use the example of wheat stripe rust in Australia. I mentioned before we had an exotic incursion in 1979. It was the first time stripe rust had ever been found in Australia. And these are photos from a colleague of mine, Dr. Colin Wellings. He took that isolate from 1979 and he put it on a set of different wheat genotypes, each one with a different resistance gene. So this wheat genotype here carries the resistance gene YR6. This one here carries the gene YR7. This is YR8, YRSP, Spalding's prolific, and this one is a gene we call YRA. Don't worry too much about these designations at this point. But there are 5 different wheats there, each one carrying a different resistance gene, and that particular isolate from 1979 is avirulent on all of those. It's causing a little bit of disease on some of them, but there's no sporulation. All right, so we would say that that particular isolate is avirulent for all 5 genes. In 1981, Colin found a new pathotype, right? So this is the same wheat lines as here, but a different rust isolate found in 1981. And you can see now that this one has acquired virulence for the gene YRA. So the old one can't infect the line with YRA, this one can. All right, so this is a new pathotype that's evolved, we believe, through a single-step mutation and acquisition of virulence for the resistance gene YRA. In 1983, he then found another variant that could take out the gene YR6. Okay, so you can see these 2 isolates of the pathogen here are avirulent on YR6. This one is sporulating quite happily on the differential with YR6. So this is a third pathotype. This is pathotype 1, pathotype 2, pathotype 3. This is the first mutant he found, and this is the second mutant that he found. And then he found one in 1986 with virulence for YR7. You should be able to see the difference there, okay? So now YR7 has gone from being effective to ineffective. So what we've seen here over a 7-year period is the development of 3 derivative pathotypes through simple mutational acquisition of virulence, firstly for YRA, the resistance gene YRA, secondly the resistance gene YR6, and thirdly the resistance gene YR7. And each one of those pathotypes was very important in growers' fields. This one caused huge epidemics in 1981-82 in southern New South Wales in a handful of varieties that were protected by YRA. This one caused huge epidemics in South Australia in 1983 and 84 in 2 varieties, one called Bindawara and one called Milawa, that both carried YR6. And this guy here caused problems but wasn't quite so bad in 86-87 in a variety that we call Hartog, which is actually Pavon. And Pavon luckily had some residual backup resistance, what we call adult plant resistance, behind YR6 and 7. All right, so that's number 4. We've talked, I'll just recap again. Sexual recombination. What was number 2? Exotic incursion, I think. Migration within an epidemiological region. This one's mutation. I think that's it.